Welcome to Genetic Engineering Explained with me, Rex. In today's section, we are going to study about bacterial transformation, how a plasmid can be inserted into E. coli. E. coli cells are naturally not competent, which means they cannot take up a foreign DNA from the surrounding. However, these E. coli cells can be made competent to take up a foreign DNA by treating them with calcium chloride. So these calcium chlorides, they increase the competency of the E. coli, to which if you add your plasmid, the plasmid will enter into the E. coli. So we incubate the plasmid and competent cells on ice for 30 minutes and then briefly give a heat shock, incubate the culture to grow, and then we plate it on a selection media. Let us assume that this is a bacterial cell with the outer membrane and the inner membrane, the chromosomal DNA inside the cytoplasm. So the outer membrane and the inner membrane are negatively charged due to the hydrophobic forces. This forms a negative cloud over the E. coli cell. When you add the plasmid is an extra chromosomal self-replicating DNA, again that is negatively charged, and this negative charge and the hydrophobic cloud, they repel each other. So naturally, a bacterial cell cannot take up a foreign DNA because they repel each other. However, when we treat the bacterial cell with calcium chloride, divalent cations, the divalent cations, they reduce the repulsive force caused by the hydrophobic surface of the E. coli cell and the negative charge of the DNA. There are a lot of theories behind this particular aspect of neutralization. However, the best known is that they reduce the negative repulsive force that exists between the cell and the DNA. So naturally, the DNA is brought into the vicinity of the E. coli cell. Now, when we provide a heat shock briefly for 90 seconds at 42 degrees Celsius, the E. coli membranes become semi-permeable. Again, there are kind of theories to explain this. So the most accepted one is that the membrane becomes semi-permeable to let the DNA in. So once the DNA enters, we culture them for 1 hour at 37 and then we plate them on a selection media. Thank you.